Yes, everybody, very good evening all, and very warm welcome to the session. So happy and really feeling nice to meet you all back in this session. So we are doing an important session on the rapid revision of biochemistry. Okay, so as such, biochemistry is a very important subtopic for your preparation, and today we are going to revise the maximum out of it, such that you will get everything out from this topic. So this is little bit about us, and a very big announcement is out. You will be having your CSIR NET notes very soon in our Plus platform. Okay, so if at all you have not yet subscribed, please do think of subscription, and seriously I'll tell you there is a number of offers that is going on, so that will make you very easy to get into the subscription. So this is little bit about me. So I have completed my Ph.D in Biomaterials after cracking my NET, and I am associated with Unacademy to make you all to crack this upcoming examination. Right, that's great guys. So let us see our today's topic of interest. Okay, very nice Himanshi, Shalu, Natasha, very nice everybody. Come on. So this is going to be the distinguishing between the plus as well as this iconic subscription. Okay, and seriously this time the discounts are quite huge. You're going to get 49% discount on the courses. Yes, Ankita. So if at all you have not yet subscribed, I think that this is what is the maximum you can get out of it. Okay, so I recommend you to go for this 12 months. Okay, so if you subscribe for 12 months, you will get 3 months free. Okay, so the current price is 16, but post discount you will get it at 13. Okay, so you will be serving almost 44%, that is 10,000 rupees you will be serving. So you can use my code Saranya Live to get into this. So the units that I'm going to teach, it is going to be inclusive of biochemistry. It is inclusive of your molecular biology. Different modules are included. So in depth I have covered this molecular biology. So I am very confident in my things. Okay, methods, and then applied biology, and then developmental biology, ecology, evolution. Yes. Okay, so these 8 units, cell biology. Okay, right. So these 8 units I am taking up 3, 4, 5, 6, 7, 8. Absolutely. So these 8 units I am doing up with Unacademy this time. So definitely you all will be benefited. So get the edge of the preparation. So this offer is valid only till tonight. So don't miss out this. Enroll at the earliest and get the edge of the preparation. Okay. So coming up to biochemistry. Right. Yes, everybody. So you are all ready to revise biochemistry. Yesterday, how was your cell biology? Cell biology is fine? Yes? Okay, yes, Natasha. Come on, guys. We all will put hands together for biochemistry. Very rapid and quick revision. Okay. So the first important concept, it is going to be this interactions. Right. So under this interaction, major of the interaction, it is going to be yes, Himanshi. So it is going to be your no covalent and non-covalent interactions. Okay, right. Yes, Rumi. Very good evening. Come on guys, let's all make out for this first revision topic. Okay, so we are going to see about something, and then quickly we will come about this question. Okay, so we'll read the concept first. Yes, Shalu, come on guys. So we'll read the concept first. So once you are thorough with the concept, then you can easily take up the questions. So among the bonds in biology, do you see any of this ionic bond? Have you heard? Yes, Sheba. Good evening. In biology, have you ever heard of this ionic bond? No? What is the equivalent of ionic bond in biology? You have heard only about this electrostatic interaction, isn't it? Yes. So you have heard about this electrostatic interaction. Yes, Vishal. But we have not much heard about ionic bond, because ionic bond is possible only in chemistry, molecular related things, Na plus Cl minus that. There is a total sharing. Here we don't have such thing. So we have only this non-covalent interaction. So this non-covalent interaction, it is going to be electrostatic in nature. Got it guys? Yes? Okay, I hope that this is little bit, clear, I guess. Yes or no? Got it? Okay, fantastic. So this electrostatic interaction or little bit of ionic interaction, it is very, very strong. It is little bit powerful because it is involving the ions. Okay. Next, the covalent bond, it is somewhere between 60 to 600. Okay. So this bandwidth is seriously very important. So this is representing so much of energy. Okay. So if at all I have an enzyme and substrate, will I have a covalent bond? I have an enzyme and substrate, or I have an antigen and antibody. Have you ever heard that it is having a covalent bond? Is it possible for my enzyme and substrate or my antigen and antibody to react in a covalent interaction? Literally it is possible? No, not at all, isn't it? So it is going to involve only the non-covalent interaction. Why? Because majority of the non-covalent interaction, they require less energy to break it or less energy to break it. Okay, so that is why generally these non-covalent interactions are more noted among the biomolecules when compared to that of this covalent bond, or very strong bonds like ionic bonds. They are not at all much involved. Why? Because they require more interaction energy. But if you take this non-covalent interaction like your dipole-induced dipole interaction, van der Waals, hydrogen bond, they all require little bit less energy. Thereby, they are preferred over your covalent bond. Got the logic now? Why non-covalent interactions is more when compared to this your covalent bonds? Yes. What is the problem with this covalent bond? This covalent as well as this ionic bonds, right? So they require more energy. Energy requirement is ultimately more. So that is why, yes everybody, so that is why they require more things to form. So it is not so easy to break or make. Yeah, they are very, very strong. Okay, yes. But if it is going to be this non-covalent interaction, so what are these non-covalent interaction? Like you see with your electrostatic interaction, let you see your hydrogen bond, let you take up your van der Waals. Van der Waals is only cloud of interaction, right? Yes. So these things, hydrophobic interaction, right? So these are all coming under non-covalent bonds. They are pretty weak in nature. Even though they are weak in nature, it is easy to make and easy to break. So you prefer them over the other. Got it, guys? Is it clear? Yeah? Okay. So if you take this van der Waals, this requires the least energy. Okay. So van der Waals, they require the least energy. Dipole induced dipole is there, but still our conventional things, your van der Waals, it is requiring least energy, followed by this hydrogen bond. Then only this covalent bond is coming. Okay. So let us take a question. The interaction energy between the atom A and B, it is 400 kilojoules. So what is the interaction between them? Is it strong or weak? This is a CSIR PYQ only. This is a PYQ. Okay. Previous year question. Okay. Previously asked question. What do you think? Okay. So what is going to be this? It is weak or strong? 400 kilojoules. So is it possible? Come on guys. Is it possible? Definitely not. Isn't it? So it is not a non-covalent interaction. So what kind of interaction is this? Absolutely. It is possible that it can be only the covalent. Yes, Himanshi, Preetham, fantastic. It is going to be the covalent. Yes, Shimpi, absolutely correct. This is going to become like a covalent interaction. Van der Waals is very, very less, 0.08. Okay, so this covalent is 400. It is going to be very, very long, high, strong bond. Yes, Deblina, Shalu, correct. Yes, Simpi, Preetham, Himanshi, come on everybody. But if I take my Van der Waals, it is just 0.8. If it is going to be a hydrogen bond, I think it is around 2. Yes, so hydrogen bond is very less than 50. Okay, so dipole is going to be, yes, Rumi. So these are all are less bonds. So if I have to put them in order, you can say that covalent and then this ionic, they are going to be very strong. So it begins with your dipole and induced dipole. Okay, so dipole and then induced dipole. Followed by whom? Followed by Van der Waals. Then it is going to be dipole. Then it is going to be your hydrogen bond. Then covalent. Then only ionic. So among them, who is weak? Dipole, induced dipole is very, very weak. Followed by Van der Waals. Followed by dipole. Followed by hydrogen bond. Covalent. Who is the strongest? It is going to be this ionic bond. Yes, everybody in this class, did you get to know about this information? Seriously, it is very important for an examination. So far, many match the following type of questions have come from this aspect. So you have to know how this interaction is taking place. Yes, hydrophobic is also very, very weak bond. Yes, Vishal. So hydrophobic is also coming. It is somewhere between your hydrogen bond and your dipole. Here it comes, hydrophobic interaction. It is also a weak interaction. Okay, right. And who is going to stabilize your DNA? DNA, we all know that it is made up of your hydrogen bond, right? But who will stabilize your DNA? Who will stabilize your DNA? DNA, it is made up of your hydrogen bond, but who is going to stabilize your DNA? It is going to be the hydrophobic interaction. Hydrophobic interaction is only going to stabilize the DNA. Very, very important question to be noted. Okay, right, we all immediately think of hydrogen bond. No, hydrogen bond they form, but who or which interaction is going to stabilize your DNA? It is going to be hydrophobic and Van der Waals. Hydrophobic and Van der Waals, they only stabilize your DNA. Okay guys, so is it clear? Shall we make up a move? Okay. So among the different states of matter, you have solid, liquid, gas and plasma, which has high entropy. Which has high entropy. Entropy means it is going to be the disorderliness. Who is that with highest entropy? It is going to be the solid, liquid, gas or plasma. Yes. So if only I gave you solid, liquid, gas, you can say gas. What about plasma? Plasma is also disordered only. Got it? Yes. So among, if I consider only solid, liquid and gas you can consider. Yes. But if I include plasma also, plasma is also highly disordered only. Okay, so this is still more, this is going to be higher. Okay, so if I take all the four options, then highest dystrophy, it will be plasma, followed by the gas, followed by the liquid, followed by the solid. Solid is it is arranged structure. Okay, so you see the crystal structure of a diamond, it is going to be highly arranged. Okay, so coming to the next question, and based on this question we are going to learn the aspect, so don't worry all. Come on. So the peptide bond. Peptide bond is formed between which one? Which residues will form the peptide bond? It is between carbon and carbon. Peptide bond, first you tell me, what is a peptide bond? Peptide bond is formed between carbon, nitrogen, carbon and oxygen, carbon and hydrogen. So which is a peptide bond? First you answer this question, then we will take up this fellow. So peptide bond is formed between your carboxyl amino acid. No, it is going to be a carboxyl group and the amino group. So it is between your C and the N, isn't it? Yes, absolutely correct. And this peptide bond is going to be very much rigid. So how is going to be the peptide bond? Yes, Shiba. Yes, Havinder. Yes, Vishal, Rumi, Simpi. Very good, guys. Yeah, so this is going to be rigid. Why it is rigid? Because it is a single bond? No. Yes, everybody. Correct, Shalu. It is going to have a partial double bond. That is why it is very, very rigid. It is going to have this omega. Omega is the angle that is formed between C and N. It is a partial double bond. This only makes them rigid. Okay. So peptide bond is planar. True or false? Peptide bond is planar. True or false? Come on, guys. Yes, everybody. True or false? I am saying that peptide bond, it is going to be, yes, peptide bond, it is going to be planar. True or false? Come on, guys. Yes, everyone. Absolutely true. Fantastic. Peptide bonds, they are planar in nature. Okay, right. So they all come in the same plane. It is going to be planar in nature. Okay, yes, absolutely. So peptide bond, it is going to be planar in nature. Okay, so you cannot change the plane. They all move in a single plane. Okay, so you have this pi angle, you have this psi angle, and you have this omega angle. This omega angle is only going to form your Cn. The Cn is only going to be this partial double bond. Okay, that is only making them to be rigid. Okay, so what do you mean by this trans confer configuration? Okay, first I'll tell you what is the difference between the term configuration and conformation. Okay, so this is even a very big important concept that you must know. Okay, so I am saying that my molecule, okay, so it is going to be in chair form. Okay, you have heard of this? Chair, boat, twist. Okay, so I am saying that my molecule, it is in the chair form. Is it a conformation or is it a configuration? What is the difference between the conformation and configuration? Come on, guys, think and answer. What is the difference between the conformation and configuration? If I say that my molecule is lying in a chair, it is a conformation or configuration? This chair, it is what is called as conformation. That means that without breaking any bond, it can form the boat. Without making any changes in the structure, it can form the twist. It can form the planar. Okay, planar, it is going to require the most energy. So this is going to be least stable. Okay, right. But if it is going to be in the chair, how is the chair? It is going to be very comfortable, right? So likewise, for your molecule, this chair conformation, it is going to be very, very more stable and least energy. Okay, I'll repeat this concept in one more slide such that you don't get tricked. Okay, so planar. If I take planar, it is called as a conformation. So what do you mean by conformation? So that means that without breaking a bond, you are going to change them to this boat. Without breaking them, you form this chair. Okay, so this chair is going to be the most stable conformation. But what is a configuration? Configuration, it is like your TV. It is like your computer. Can you change it instantly? No, you have to change the equipment only. Correct. You cannot change the configuration. But you can download an app, you can do some modification. So that is all minor changes is confirmation. But configuration you cannot change. Got it? Okay, fine. So what about the cis and the trans? Cis and the trans. It is a confirmation or configuration? Come on guys. So I am talking about the cis and the trans. It is a confirmation or configuration? Make it fast. Yes everybody. Cis and the trans. Yes Kunal. It is a configuration. Very good. Yes. Configuration you cannot change, isn't it? If it is trans, these two are oppositely located. That is why it is called as trans. If it is going to be cis, all of them will be in the same region. Yeah. So if you want to change it, you have to break this bond and you have to put them up. Okay. So this is why it is called as configuration. Got it? So now you got the difference between the confirmation and this configuration. Right? So cis and the trans, they are going to be the configuration. Majority of your amino acids, they appear in cis or trans. Which form? Be quick. Majority of your amino acids will occur in which form? Cis or trans? Majority of your amino acids are going to appear in which forms? It is going to be cis or trans. Very good. Yes Kunal, Vishal, Kavya. It is absolutely going to be your trans form only. Trans is major. One fellow is there. He will be in the cis. Who is that? Yes, simply. Yes, Takoti, Methila, Shalu, correct. Himanshi, correct. Yes, Vishal. Trans will minimize your steric hindrance. Which fellow will be in the cis? Our own proline, isn't it? He is always the spoiler, no? So he is in the cis because he will give a steric hindrance. He doesn't allow anybody to come near him. Yes, that is why he is standing in the cis conformation, but not other amino acid. Okay, so now why a peptide bond is rigid? Because of this CN double bond, partial double bond. You can't call it as a double bond, but CN is going to be a partial double bond. Okay, so this is going to be about this omega. What is about this psi? I have told you that psi is going to be a boring angle. Which is boring? Between C and C, same type, isn't it? Yes, if it is between C and C, it is very much boring, same sound. It is psi. Okay, so if it is between C and N, it is omega. Okay. And what is going to be your pi angle? What is going to be your pi angle? C and NH. Okay, C and NH is pi. And what is this C double bond O and N? C double bond O and N, it is going to be omega. Okay, this is highly electronegative. Okay, so they will create this pi interactions and all. Okay, so that is why this is going to be omega. But between your NH and C, it is going to become your pi angle. Okay, so NH and C, it is going to be pi angle. C double bond O and N, it is going to be your omega. So minor points only, but definitely you have to make a thing. Okay. So we will take up the next question, and with the help of the next question you will be able to revise and do justice. Shall we take it up? Come on guys. The activity of an enzyme, it was measured. How it was measured? By varying the concentration of the substrate S in the presence of 3 different concentrations of inhibitor. It is 2, 0, 2 and 4. So first inhibitor concentration is 0. What does it mean? What does it mean? It is no inhibition. Isn't it? Right. Fine. Yes everybody. and then it is going to be 2 and 4. It is increased inhibitor concentration. So now this reciprocal blot, it is showing the graph like this. Okay, yes everybody. So this reciprocal plot, it is going to show the graph like this. So what is the type of inhibitor? It is substrate inhibition, uncompetitive, mixtured or competitive. Very good guys. Yes Rumi, yes Kunal. It is going to be a competitive inhibition. So always we make clues to remember this. Yeah, okay. So if it is going to be competitive inhibition, I told you the clue CX. Yes. So if you compare your inhibitor with your non-inhibition, yeah, it is going to be in the X shaped. So if it is X shaped, it is competitive. And what about your uncompetitive inhibition? It is parallel. U is parallel, uncompetitive is parallel. And what about your non-competitive inhibition? Non-competitive inhibition will be V-shaped, NV. Okay, so if you remember these 3 clues, absolutely it will be helpful to you. So competitive inhibition will look like X shape, exactly how it is given here. Okay, it is going to be X shaped. And uncompetitive is U parallel, and then non-competitive it is going to be the NV. Got it guys? So everybody is clear about these points. Yes everybody. So it is going to be CX, your U parallel, and then this NV. It's a very typical question usually that is being asked up with. Okay. So coming to the next question as well as the concept to revise. Okay. So if at all my amino acid is going to be alpha helix, what are the amino acids that should be present in an alpha helix? Make it fast. If at all it is going to be in an alpha helix, what are the things that should be present? You remember this clue? Malic. Yes? Yeah. So this is going to be malic. Okay. So what it should avoid? It should avoid proline. Why? Proline is a toppler. You don't want him. So wherever you see proline, you will avoid. What about glycine? Glycine also you avoid. Why? Yes. So why you avoid glycine? Yes, malric we made. So why you glycine you avoid? It is very flexible. So if it is going to be too much flexible, then also it will not form a proper helix. Right? Yeah. So it is going to be a helix breaker. Yes, it is called as a helix breaker. Okay, right. So it is avoiding, it is a helix breaker. Who are the helix breakers? Proline and glycine. And helix formers, it is going to be your methionine, alanine, leucine, arginine, glutamic acid and lysine. So it is going to contain a mixture of charged and uncharged, isn't it? So these are all non-polar. Okay, so this is our polar. Okay, because you remember our helix is used as a transmembrane protein. So they will have some hydrophobic region also. Outside it should be hydrophilic region. Okay, so that is why this is sounding like this. Okay, but in this middle it will be hydrophobic. At the end maybe it is hydrophilic such that it will survive both the environments. Okay, so which of the following will take up? Definitely you will go for D. Why D? Because there should not be any glycine or proline. By that elimination only you can come up with this. Okay, so I think this question we were discussing in the morning plus class or special class, I forgot. Okay, right. So we were discussing about this, how many number of bonds they can form, and we were also discussing who has the maximum possibility. Yes, and also we were talking which is having the largest possibility of hydrogen bonding. Okay, so it is adenine or guanine or cytosine or uracil. Very good everybody. Yes Tripadim, yes Vishal. Keep it up guys. Yes Himanshi. Good. Fantastic. Too good ma. All of you rock like anything. Yes, it is going to be none other than guanine. Why so? This is because of the fact that, yes, Rumi, Shima, Shalu, Kunal, Tripadim, Himanshi, because, yeah, if you take triple bond, even your cytidine can form triple bond. Okay, but among them, this guanosine is having an advantage. What kind of advantage it is having? It is having, yes, Kunal, correct. It is having one acceptor and it is having two of the donors. But what about your cytosine? Yes, it has two acceptor. Isn't it? Two acceptors it is there. Only one donor is there. Yeah, correct, Kunal. We did it in scientific, your biochemistry course. You remember that? Good. So this is why they have a largest possibility of having your hydrogen bonds. Guanine is only going to have the largest possibility of hydrogen bonds. Okay. So glucose residues in amylose. It is linked by which of the following bonds? Amylose. It is linked by which of the following bonds? Come on guys, everybody in this class, make it out, make it out. Yes all. Let's make it out for this. So, very good. Yes, Kunal. Come on, what about others? Amylose. First you tell me the outliners. Right. Very good, Vishal. Havinder. Very good, Havinder. Yes, Tripatim. Fantastic. So, this is all going to be linked by, yes, Himanshi, alpha 1,4 linkages. Okay, so amylose, it is going to be alpha 1,4. Yes, Sarita, it is alpha 1,4 linkages. But what are the outliners? Correct, A for alpha, correct, absolutely. So, what is for beta? Beta 1,4 only few will have. Do you remember? Yes. A for amylose, right. So, amylose, amylopectin, right there. Only outliners you remember. Beta for cellulose, beta for lactose. Apart from that, all are chitin. Apart from that, all are going to be alpha only. Okay. So, cellulose, lactose, chitin. It is only having this beta 1,4. This is the reason no enzyme is available to degrade the cellulose. This bond is very difficult to break. Yeah, because none of the enzymes, they have the capacity to break them. So, beta galactosidase, it can act on lactose. Chitin, these are all very, very strong. So, that is why your bacteria prefers the glucose over the galactose. Right? Because this lactose is also having this beta 1,4. But what about the amylose? Amylose is going to have the alpha 1,4 link. Okay, so alpha 1,4 link it is going to have. Okay, C1 carbon with the C4 carbon. That is what is going to be forming this amylose. Okay, what about this amylopectin? Amylopectin, it is branched. Okay, so apart from this alpha 1,4, it is having this alpha 1,6. This is the branch. Amylose only straight chain. There is no branches. Cellulose beta 1,4 but no branches. Yes, but if you take this amylopectin in the plants, it is going to show some branches. And what about this glycogen? Glycogen, it is present in the animal, isn't it? Storage part. Yes, Sarita. So it is going to be your alpha 1,4 and 1,6. But see the frequency. Here it takes place for every 20 subunits. Here very, very near it takes place. That is why it looks like this. This little bit, no space is there. Here it is very, very clunky. Why? Because within 10 subunits it is going to form a branch. Yes? Shall we recap? Shall we recap this one? You are only going to tell me. Okay. So amylose, amylopectin, cellulose, glycogen. Amylose, it is alpha 1,4. There is no branch. Amylopectin, alpha 1,4, alpha 1,6, branch is there. Cellulose, beta 1,4, no branch. Glycogen, alpha 1,4 and alpha 1,6, branch is extensive. Branch is extensive. Okay, if it is like for every 10 residue, if it is for every 20 residue, this will be for every 10 residue. So this is have an extensive branching. That is what you see up here. Got it, guys? So is it all clear like how it works? Yes? Okay, fantastic. Now let us all take a question. We will relaxly read this question and you will be able to grasp this concept as well. So you have 4 single amino acid mutants. They are A to D. It is having a protein in this epitope region. Okay. So now what you are doing, you are making a monoclonal antibody and it is expressing the 4 proteins. Okay, so listen carefully. This is a little bit lengthy question, but still it will be of great help in your examination. So slowly and carefully you have to develop the habit of reading the questions and responding to them. It is an art of learning to read a question patiently and answer. Shall we all try with this question? You all will do this with me? Come on guys, let's try out. So, whenever you are doing this SDS page, okay, and then you are making the bands to be transferred to this nitrocellulose membrane and this western blot using your monoclonal antibody, okay, so this is raised against your wild type. These images are got, okay. So, A, it is getting the same one for your control also, for your new one also. B, this is getting double band. C and D, it is absent, right? Fine. So this is the wild type. Okay. So this is what you get. So this 4 single mutation upon sequencing, it was found to be number 1 is valine to alanine. Number 2, glycine to proline. Number 3, alanine to aspartic acid. Number 4, isoleucine to leucine. Okay, got it? Right? Fine. So now, before taking up these options, shall we just do a classification very quickly for our reference? What are these non-polar aliphatic amino acids? Non-polar and aliphatic amino acids. What are they? You remember the clue? Gap will met. How many of you remembered it? Glycine, alanine, proline, valine, isoleucine, leucine, methionine. Gap will met. Yes? Fantastic guys. So with the clues you can make a fantastic memory. Yes, it is going to be the gap will met. Okay. It is non-polar aliphatic. Okay. The next one, it is non-polar and aromatic. So what are they? It is the wife. Tryptophan, tyrosine, phenylalanine. Okay. Next it is going to be polar and uncharged. Who will come under this polar and uncharged fellow? Yes, it is going to be mine, gine, TCS, asparagine, glutamine, and then Tyrosine, cysteine, and then serine. Mine, gyne, TCS. Then positively charged, yes, HRK, histidine, arginine, lysine. And if it is negatively charged, glutamic acid, aspartic acid. Okay? So, will you be able to remember all this classification by heart? Shall I ask you the question? Will you tell me true or false? Yes? How many of you are ready for this? You want to read this? Just read it for a moment. I am going to change the screen quickly. Memorized? Memory is over. Shall I ask you questions? Yes or no, guys? So, non-polar aliphatic gap element, non-polar aromatic wife, polar uncharged mine gyne TCS, positive HRK, negative is D. Ready? Shall I ask you questions? Then only you will be able to answer this question. Okay. So, tell me about cysteine. Cysteine, it is polar or non-polar? Cysteine, it is polar or non-polar? Type P or N? Cysteine, it is polar or non-polar? Make it fast, make it fast. Am I there, guys? Yes? Only one answer came. Yes, Vishal. Yes, Kunal. Amrita, Tripadim, Shalu, Shiba, Himanshi. Cysteine is uncharged. I agree that cysteine is uncharged, but it is polar. Okay. Cysteine is polar. It has SH group. No. It is polar. Okay. Cysteine is polar. Okay. Yes. Polar and uncharged. Correct. Yes, Himanshi, Rumi, Shushmita, Kunal, Shayani. Yes, everybody. So, what about tyrosine? Tyrosine, polar or non-polar? Tyrosine. Be quick. That's all. Tyrosine is going to be tyrosine is going to be non-polar only. Aromatic non-polar. Okay. So, who will come under the polar? That is why I told you. I just want you to tell this one more time. Okay. I'll just tell you one more time. See here. Your polar is going to be mine gyne TCS, HRK D. That's all. All others are non-polar. Got it, guys? One more time I'll just give you this. Yes? So, if you take this non-polar, who are all coming under non-polar? Either of these you learn. Okay. So, under non-polar, it is going to be your gap, will, met, and then wife. Okay. This is non-polar. This is going to be uncharged. This is what is called as hydrophobic also. Okay, right? Yes. If it is going to be polar. Okay, yes. So, under this polar, you have two categories. One, it is going to be the polar charged. Under that, you have positively charged, that is going to be HRK. You have negatively charged, that is going to be your aspartic acid and glutamic acid. Got it? Yes, everybody in this class, did you get it now? Yes? Okay. And in the meanwhile, if it is going to be uncharged, it is mine, glutamine, gyne, asparagine, and then TCS. TCS is going to be your cysteine, serine. And what is this T? Threonine. This is not tyrosine. This is threonine. Okay? Right? Fine. Yes. So, wife is tryptophan, tyrosine, phenylalanine. So got the difference between these two? Yes, everybody? This is very, very important. Okay, if you know this only, you will be able to answer this question. Okay, right. So now you tell me valine, alanine, both of them are synonymous mutation or non-synonymous mutation? Yes, Rishabh. Valine and alanine, it is going to be synonymous or non-synonymous. Synonymous means similar. Valine and alanine, valine is also hydrophobic or it is non-polar. Yes. So what about alanine? Yes, alanine is also going to be non-polar. Yes, so it is all similar. So this is going to be your synonymous. Got it? If it is going to be similar, it is what is called as synonymous. Okay. Coming to the next one. Yes, it is going to be glycine. Yes, to proline. Okay. So glycine to proline. Yes, at least they are non-polar, little bit. Okay, but not exactly like your valine to alanine. Okay, so they are look-alike. But this fellow is very flexible in its property. This is very rigid in property. Okay, so apart from that, yes, this is not exactly matched like this fellow. Okay, because property wise glycine is very, very flexible. We all know that glycine is flexible, but what about this proline? It is too much rigid. Even though they are the non-polar fellow, proline is always mischievous, isn't it? So set him aside. Now let you take the third one, alanine to aspartic acid. Alanine is non-polar. Yes. So what is the nature of alanine? Alanine is non-polar. Aspartic acid is polar. So this is going to be fine. No, this is non-synonymous. Isoleucine, leucine, it is similar. Correct. So this glycine and proline, even though they are both are going to be hydrophobic, but if you take about the property, it is becoming different. Okay. So C and D, this monoclonal antibody, it is not binding. Why it is not binding? Because it is going to be different. That is why it is not binding. In A and B it is binding. Why it is binding? Because it is going to be similar. That is why it is binding. So shall we check out? B is due to valine to alanine. B, it is due to valine and alanine. That is why it is getting a band. It is right. C, it is due to glycine and proline. Maybe this is also right because it is non-synonymous. B, it is due to glycine to proline. So this is not possible. D, it is due to valine to alanine. Not at all possible. D, it is due to isoleucine to leucine. No. A is due to your alanine to aspartic acid. Not at all possible. C is due to valine to alanine. No, it should have got a band. And this is missing band. And sorry, this may be right. So this A may be I2L, this may be right, but this statement is wrong, right? So that is why only both the things correct, it is going to be your option A. How many of you understood this aspect? Did you get the point? Like why a band is formed here and why a band is not formed here? What could be the reason behind it? Everybody in the class, did you understand the classification? One more time, shall I ask you the classification? Yes, you will all be able to understand. Okay. So, isoleucine is non-polar. True or false? Isoleucine is non-polar. True or false? Fantastic. Yes, everybody, isoleucine is non-polar. Lysine is polar. Oh, Pavan, that's okay. You can see the recorded version. Don't worry. It is a true statement. Serine is polar. True or false? Yes, Shiba, Amrita, Shalu, Shushmita, Chetna, Shiba, Amrita, Kunal, Himanshi, Tripadim. Very good. Serine is uncharged polar. Okay. Yes, Amrita. Serine is uncharged polar. Right. And this lysine is charged polar. So charged polar is what is called as hydrophilic. So uncharged polar is also called as hydrophilic. Okay, right. So if at all my protein, okay, it is having hydrophobic domain, okay, right, it is going to contain option A, lysine, arginine, serine. Option B, glycine, alanine, valine. Option C, threonine, cysteine, serine. Option D, it is going to be your aspartic acid and your glutamic acid. So my protein is having a hydrophobic domain. So which option should be perfectly matching with this condition? Hydrophobic domain. My protein is going to contain the hydrophobic domain. See that I have jumbled the options. A, B, C, D have written in different order, so don't get tricked in this order. Yes. Maybe that could have tricked you. Yes. Yes, Kunal. Pavan, Tripadim, Shushmita, Himanshi, Amrita. Fantastic, everybody. Yes, Pavan. You got it. So this is going to be your gap will met and then wife. Yes, Shaluk. Gap will met and wife. Yes, Chetna. Absolutely correct. So this gap will met and wife. It is going to be, yes, Rumi. Fantastic. It is non-polar. So non-polar and uncharged is only going to be hydrophobic. Okay, so they are only going to be hydrophobic, none other than them is going to be hydrophobic. Okay, so I hope that this question is clear. So in one more session, please, we will make out to revise your metabolic things. Okay, and this fellow is often coming up in the exam. So let us take this question before we sign off from this topic. Yes, okay. So a tryptic digest of heptapeptide. Heptapeptide means how many residues are there? It is a 7-mer. Okay. It is built using 3 lysine, 2 alanine, 1 tyrosine and 1 phenylalanine. Okay. So this yielded tri and tetrapeptide. So this yielded a tri as well as a tetrapeptide. Okay, right. So which of the following is the correct sequence of the heptapeptide? Yes. Fantastic, guys. So after you use the trypsin, it is going to give you a tri. Very good. Yes, everybody. Come on, Rumi, Shayani, Shalu, Tripadim, Shushmita. Correct. So which of the following is going to be correct? So where does this trypsin digest? Trypsin, it is going to be an endonuclease. It will cut at N terminus or C terminus? Where trypsin will... okay, Anjali, don't worry. Yes, trypsin, it is going to be an enzyme. It is an endonuclease enzyme. It will cut at the C terminus of arginine and lysine. Okay. So what is the difference between C terminus and N terminus? For example, okay, so this is 1, 2, 3. Okay, so imagine that this is going to be the lysine. Okay, right. If it is going to be cutting at this C terminus, it will cut here. If at all there is an enzyme that is going to cut at N terminus, it will divide this into 2 plus 3. Now it is dividing it into 3 plus 2. Got the difference between the N terminal cut and the C terminal cut? Everybody in this class, did you understand what is an N terminal cut and what is a C terminal cut? Any doubts, please let me know. Is this clear? Yes. So N terminus means, see, this is going to be the third residue. This side N is there, this side C is there. So at the C only it will cut. So it is going to cut here. So it will form 3 and 2. Okay, right. Yes, absolutely. Yes, correct. So second rule is that there should be no proline nearby. Okay, so there should be no proline nearby. Third one, it should not be at the end. It should not be at the end. So you want them to cut such that it is going to form a tri and a tetra. Tri means 3. Tetra means 4. Okay, right? Fine. So if you take this, can this be cut? Is there any possibility that here it will be cut? This is the first bond. So first means it is an exo. So your molecule cannot act at this. Can this cut here? Yes, it can cut here. Can it cut here? No, this is the end, no. So it becomes your exo. This is also exo. Here no bond. Okay, so here it is the beginning. Only these two are the first bonds. So in these two position it cannot cut. But here it can cut. So it is going to be 1, 2, 3, 4, 5 plus 2. Is it correct? No. So this is not my correct answer. So let us make a move towards the second option. Shall we see? Here one cut. That's all. So this is going to be 2 and 5. Is this the correct answer? Is this a correct answer? No. Check out here. 3 and 4. Is this my correct answer? Come on guys. Check out and let me know if this is going to be the correct answer. Yes. It is only going to be the correct answer. The correct answer is only going to be C. Okay. So what are the rules? Number 1, it should cut only at endo. Okay. So I'll show you an option of endo and exo. Yes, Arpita, Tripadi, Anjali. Come on. So this is going to be A. This is going to be R. This is going to be V. This is going to be K. This is going to be I. This is going to be L. Okay. Right. Fine. So this is the first bond, second, third, fourth, fifth. Okay. So identify the exonuclease site of action. Exonuclease. It is 1 and 5. It is 2, 3 and 4. C, all. D, none. Which is the site for exonuclease action? Exo means it is going to be at the end. Which one is exonuclease? Come on guys. Which one of the following is exonuclease? Absolutely. Very good everybody. Keep it up. Yes, Chetna. Yes, Kunal. It is going to be 1 and 5. This is only terminus, no. This is only going to be exo. Yes, Satya. It is only going to be the exo. So what are the endo sites? Endo, it is going to be 2, 3 and 4. And one more condition, there should be no proline. Okay. So if at all I am using my trypsin. Okay. So I am using my trypsin to digest this peptide. Okay. So what will be the fragments I get? Will I get 4 plus 2? Will I get 3 plus 3? no cut, only one full length I am going to get, that is 6. So if I use trypsin to cut this molecule, I am using trypsin to cut this molecule. So what will I get is my question. What fragment will I get? Yes, Taiba. Yes, Kunal. Arpita. Shalu. Good try. I told you that it is going to cut at endo, isn't it? And then yes, yes, Narayan. So it is going to be endo, and it has to cut only at R and K. Okay, so the recognition site is only going to be R and K. Okay, so it is going to be endo. It is going to cut at R and K. Okay, so then where it is going to cut? It is going to cut at the C terminus. So if it is going to be C terminus, it is going to cut here only. So it is going to be 1, 2, 3, 4, 4 and 2. Hence your option A alone is going to be the correct answer for this question. Got it, everybody? Why all other options are gone wrong? Because it cuts only at the C terminus. It is not cutting at the N terminus. This is the C terminus. That is why only A is right. If it is cut at the N terminus, maybe 3 plus 3. But it is cutting at C, no. So it is only 4 plus 2. It cuts at R and K. Trypsin will cut at R and K. Yes, Narayan. I am going to tell you one more final time. Yes. So there are few rules for the trypsin. Number 1 rule, trypsin is going to act only at the endo site. Endo means inside. Okay, so it is not going to act at any other site. It is going to act only at the endonuclease. Endo means inside. It will not cut this final bond. This final is meant for exo. So it is not going to cut at the end. It will cut only the inside bond. If it is going to be this one, it is an inside bond. No, so it can cut. But this end, it will never cut. This end, can it cut? This KK, can your trypsin cut? Do you think that this KK, this trypsin will cut? Yes or no? Come on guys, think and let me know. This KK, will this trypsin cut? It can never cut, isn't it? Why? Because it is going to be an exo site. Your trypsin can act only at the endo site. You understood? Fantastic. So whichever is going to be the exo site, only the exopeptidases like your carboxypeptidases will only cut. Right? But as far as this trypsin is considered, trypsin is going to be endonucleases. That means it is going to cut deep inside. So it is not going to touch this part. Okay. So now I hope that everybody got clarity on this. Right? Okay, fantastic guys. I think that we have discussed few important points related to biochemistry. Definitely we'll continue on our revision in one more session. It seems to be a little bit big. If you find something inappropriate, you can always report, and it is absolutely in your hands. You have a control over this. Right, and please don't forget about this Unacademy wonderful offer that is going on. It is bigger. Yes, Kunal, we'll do that NMR. Definitely, it is in my mind. We'll do in this month of August. Don't worry. This NMR, sorry, we will definitely cover up. I think geological time scale we are done. NMR alone we are left with. Yes, so you have to use the code Saranya Live. Okay, such that you will get wonderful discounts. Got it? Yes, so don't miss out all those things. Yes, Shalu, Himansi, Kunal. Thanks a lot, everybody. Thanks a lot for joining me live. All the very best for your preparation. Fossils will do. Okay, so in the next doubt clearer session, you just remind me of the pending things such that we can cover either in the special or the YouTube such that you can have the access. Yes, Tripadim, Chetna. Thanks a lot, everybody. Thanks a lot. So have a nice day ahead. I'll meet you tomorrow with more information. Tata, bye-bye. See you all. Thank you, everybody. Bye-bye. Don't forget to use the code. Yes, next class is tomorrow, Pavan. Tomorrow same time, 5 p.m. we'll have the session. Okay, right. Bye bye all. Thank you guys. Meet you.